Okay, hello to all of you. Let's discuss chapter number 17. So, with this chapter, basically, we are starting the process of cellular respiration. So, this is a process by which the energy is extracted from the food we have eaten. So, this is the cellular respiration is an aerobic process, and aerobic process means it needs the presence of oxygen. It needs oxygen, and so basically oxygen in this process acts as a final electron acceptor. So this process is called cellular respiration. So you must be knowing what cell respiration is basically. So respiration is a process in which we inhale oxygen and carbon dioxide. So in this chapter, in these upcoming chapters, you will understand that the oxygen which we are taking in, which we are inhaling, what is its role. So basically, oxygen which we are taking in our body, actually acts as a final electron acceptor, and in this process of cellular respiration, carbon dioxide is released, and that is the carbon dioxide we are exhaling out. So this is a process by which the energy is extracted from the food we have eaten. So the energy is there in the food, but it has to be extracted, and the energy in our food is in the form of high energy electrons, and that energy in our cells is converted into ATP. Now ATP we learned in last lecture, it's the energy currency. It's called adenosine triphosphate. So this process of cellular respiration, it goes through different stages. uh, there are basically four stages. First is glycolysis, which is the focus of this chapter, and in glycolysis the glucose is converted into pyruvate, and then pyruvate is converted into acetyl coenzyme A, and then acetyl coenzyme A combines with oxaloacetate. It goes through Krebs cycle, and then the last stage is oxidative phosphorylation. So these are different stages of cellular respiration, and in this process the energy is extracted in the form of ATP. And also we have learned in last few lectures that we have some coenzymes like NAD plus and FAD. These acts as the electron carriers. They are the ones which carry the electrons, the high energy electrons from the food. So and they are the ones which carry these electrons, and when they carry these electrons, they themselves get reduced, because the gain of electrons is called reduction. So they themselves get reduced, and then they eventually become oxidized themselves when they deliver these electrons to the destination of these electrons, which is the inner membrane of mitochondria. Because in the inner membrane of mitochondria there are some proteins, and so these electrons, they flow through these proteins, and eventually they are accepted by the final electron acceptor, which is oxygen. And oxygen finally accepts these electrons, and when the oxygen accepts these electrons, it itself gets reduced and it forms water. So in this chapter we are going to focus on the first stage, which is glycolysis, and then in other upcoming chapters we will be talking about the other stages. So this is in chapter number 17 we are going to talk about glycolysis as well as fermentation. So something important to know about glycolysis is a process which takes place in the cytoplasm, and this process can take place in the absence of oxygen also. Okay, so in this process, let me tell you first of all, so this is the journey of one molecule of glucose. So basically the cellular respiration process, when we study, we study this whole process by taking one molecule of glucose. So we talk about what is the journey of one molecule of glucose, through how many stages it goes through, and in every stage how many molecules of ATP are produced and how many coenzymes they get reduced and all that. So basically in glycolysis, it takes place in cytoplasm. It does not need oxygen, and there is an net output of 2 molecules of ATP. Only 2 molecules of ATP are produced in glycolysis, and there are 2 molecules of NAD plus which get reduced to 2 molecules of NADH. So this is the overall outcome of glycolysis. So So I'm going to go over one slide where the whole process of glycolysis is described, and I'm going to explain all the 10 reactions of glycolysis through that slide. And you can go over all the other slides because all the information I'm going to give there is there. So what is important for you guys to know, the most important thing for you guys to know is the outcome of glycolysis, and also you need to know the reactions of glycolysis, and in all reactions what is happening and which enzyme is involved. So all that information you need to know about glycolysis. Now what does this slide shows us? This slide shows us that glucose, when it enters into glycolysis, it's converted into eventually into two molecules of pyruvate, and there is here in so glycolysis can be actually split into two parts. In the first part, ATP is actually used. You can see ATP is getting converted into ADP, and then there is a formation of 4 molecules of ATP. So there is net outcome gain of ATP is 2 molecules, and 2 molecules of NAD plus they converted into 2 molecules of NADH. Now this pyruvate, after that, which will be following in other chapters, this pyruvate can this pyruvate can be further converted into acetyl coenzyme A, and then acetyl coenzyme A enters into citric acid cycle, and then oxidative phosphorus. So this is the whole path of cellular respiration or aerobic respiration. Now if there is no oxygen present, as you know that in the cellular respiration the final electron acceptor is oxygen, so this is aerobic process. If oxygen is not present, then this pyruvate can be converted into lactate by lactic acid fermentation, or it can also be converted into ethanol by alcoholic fermentation. So we're going to talk about these things also in detail. So this is basically showing you the reactions of glycolysis, and this is the description. I'm going to explain you from this picture. So okay, so if we eat 1 molecule of glucose, then what happens to the glucose molecule? So in the first step, glucose is converted into glucose 6-phosphate. So you can see that in this the glucose is getting phosphate. Now from where this phosphate is coming? This phosphate group is coming from ATP. ATP is adenosine triphosphate. So ATP gets converted into ADP and glucose gets converted into glucose 6-phosphate. So the phosphate is given by ATP. And this step takes place with the help of the enzyme called hexokinase. Then in the next step, glucose 6-phosphate is converted into fructose 6-phosphate. Now glucose and fructose, they are isomers. So this step takes place with the help of the enzyme phosphoglucoisomerase. So here basically they are just glucose and fructose, they are getting converted into each other, they are isomers basically. Then fructose 6-phosphate gets converted into fructose 1,6-bisphosphate. So which means there is a addition of one more phosphate. So here is 1 phosphate, in this compound there are 2 phosphates. So these are all intermediate metabolites. So from where does this second phosphate is coming? This second phosphate is again coming from ATP. So ATP is giving this phosphate and it's getting converted into ADP. And this reaction takes place with the help of the enzyme phosphofructokinase. Now something very important to know here is on this step and this step, ATP is being used and ATP is giving phosphate group, and ADP is being ATP is getting converted to ADP. So in these reactions actually the cell has to actually invest the energy which is in the form of ATP. So these steps or this phase of glycolysis is called energy investing phase. Okay, here your cell is actually spending energy for the glycolysis. Now here fructose 1,6-bisphosphate is converted, is actually, this has 6 carbons. So this is actually going to be cleaved here and it is going to be converted into 2 compounds. One of them is called dihydroxyacetone phosphate, another is glyceraldehyde 3-phosphate. So each has 3 carbons. So here fructose 1,6-bisphosphate or intermediate metabolite fructose 1,6-bisphosphate, which has 6 carbons, is going to be cleaved, or it is going to be converted into 2 compounds which has 3 carbons each, and this reaction takes place with the help of enzyme called aldolase. Now glyceraldehyde 3-phosphate and dihydroxyacetone phosphate, they are also isomers. Okay, they are also isomers, and the dihydroxyacetone phosphate is all converted into glyceraldehyde 3-phosphate, and this takes place with the help of the enzyme called triose phosphate isomerase. So they are isomers, they are interconvertible, they can be converted into each other. So dihydroxyacetone actually gets converted into glyceraldehyde 3-phosphate. Now glyceraldehyde, so it means that at this point there are two molecules of glyceraldehyde 3-phosphate. So in this slide, everything is shown with one molecule of glyceraldehyde phosphate. So whatever is seen beyond this point has to be multiplied by 2. Glyceraldehyde 3-phosphate is going to be, in the next step, it's going to be converted, glyceraldehyde phosphate is, in the next step, is going to be converted into 1,3-bis-phosphoglycerate. Now what is happening at this phase? So this glyceraldehyde 3-phosphate, it is going to be converted into 1,3-bis-phosphoglycerate. Here this glyceraldehyde 3-phosphate is getting oxidized, which means electrons are going to will be taken from this intermediate metabolite, and are these electrons going from this intermediate metabolite, and NAD+ is going to be reduced and converted into NADH, and this takes place with the help of an enzyme called glyceraldehyde 3-phosphate dehydrogenase. And inorganic phosphate is being added, and this compound is getting converted into 1,3-bis-phosphoglycerate, which means it has 2 phosphates. In the next step, 1,3-bis-phosphoglycerate is going to be converted into 3-phosphoglycerate. So 1,3-bis-phosphoglycerate has 2 phosphates and 3-phosphoglycerate has only 1 phosphate. So where is the second phosphate going? This phosphate is going to be given to ADP. It's going to be given to ADP, and ADP will be converted into ATP, and this takes place with the help of the enzyme called phosphoglycerokinase. So 1,3-bis-phosphoglycerate is getting converted into 3-phosphoglycerate, and this 1,3-bis-phosphoglycerate is giving its phosphate to ADP, and ADP is getting converted into ATP, and this is taking place with the help of the enzyme. So this process, so here is molecule of ATP which is produced in the process of glycolysis. So remember that there were 2 molecules of glyceraldehyde 3-phosphate, so which means that there will be 2 molecules of ATP. So this process by which ATP is produced here is called substrate level phosphorylation. So substrate level phosphorylation is a process in which ATP is produced from ADP, and the phosphate group is given by some intermediate metabolite, and this process takes place with the help of the enzyme. Now 3-phosphoglycerate is then converted into 2-phosphoglycerate. So this takes phosphoglyceromutase. 3-phosphoglycerate getting converted into 2-phosphoglycerate, there is only change in the position of the phosphate group. Here the phosphate group is on the 3rd carbon, here the phosphate group is on the 2nd carbon. Then 2-phosphoglycerate is then converted into phosphoenol pyruvate, and this takes place with the help of the enzyme called enolase. And then in the last step, phosphoenol pyruvate is converted into pyruvate. So phosphoenol pyruvate, which has the phosphate group, this phosphate group will be given to ADP, and ADP will be converted into ATP. So this takes place with the help of the enzyme pyruvate kinase. So this is another step where ATP is produced by the process of substrate level phosphorylation. So again, substrate level phosphorylation is a process by which ATP is produced from ADP, and the source of phosphate group is some intermediate metabolite, like here the phosphoenol pyruvate, it has a phosphate group. So this phosphate group will be given to ADP, and it will be converted into ADP. ATP from ATP will be formed from ADP, and this takes place with the help of the enzyme called pyruvate kinase, and eventually this compound, this intermediate metabolite, will be converted into pyruvate. So remember that after this step everything has to be multiplied by 2. So in the process of glycolysis, you start with 1 molecule of glucose and you end up with 2 molecules of pyruvate. So glucose has 6 carbons, pyruvate has 3 carbons. So basically this step is called glycolysis, because here actually the sugar is, the glucose is actually lysed. So glucose has 6 carbons, pyruvate has 3 carbons. So from one molecule of glucose, 2 molecules of pyruvate are formed, and in this process 2 molecules of ATP are used here, and in the second phase 4 molecules of ATP are produced. So the net gain of ATP is 2 molecules of ATP, and here NAD plus is getting converted into NADH. Multiply by 2, there are actually 2 molecules of NADH which are the 2 molecules of NAD plus which are getting reduced to 2 molecules of NADH. So this is glycolysis. Now if the oxygen is present, then this pyruvate will be converted into acetyl coenzyme A, then it enters into Krebs cycle. That's what we are going to study in the upcoming chapters. But suppose if there is no oxygen present, then this pyruvate can undergo fermentation. Now there are two kinds of fermentations. So this is basically showing from glycolysis pyruvate is formed. If oxygen is present, then this path. Okay, this is a path we are going to follow in the upcoming chapters. If oxygen is not present, then this pyruvate can undergo fermentation. There are two kinds of fermentations. One is lactic acid fermentation, another is ethanol fermentation. In lactic acid fermentation, pyruvate is actually converted into lactate. So here basically this pyruvate, so in the glycolysis we just talked about that there are 2 molecules of NADH they are produced, right? From NAD plus to NAD plus carries the electrons and it gets converted into NADH. So this NADH which is produced in glycolysis, it needs to be regenerated back to NAD plus, because otherwise the process of glycolysis will stop. So it's very important for NADH to get converted back to NAD plus. So which means that NAD plus, when it gets converted into NADH, it is actually reduced. So this NADH needs to give back its electrons. So this NADH actually give back its electrons to pyruvate, and this pyruvate gets converted into lactate. So when the pyruvate, so this is the pyruvate, so when it accepts the hydrogen atoms, because electrons are in the form of hydrogen atoms, so when pyruvate accepts the electrons or hydrogen atoms from NADH, you can see this is your lactate. So it has been formed from the pyruvate after accepting one H here and one H here. So pyruvate gets reduced because it accepts the electrons. NADH gets oxidized and it gets converted into NAD plus, and it's very important for NADH to be regenerated back to NAD plus. Now the other kind of fermentation is called alcoholic fermentation or ethanol fermentation. So in this step, pyruvate is first of all converted into acetaldehyde. So pyruvate has 3 carbons, acetaldehyde has 2 carbons. So where is the third carbon going? It is released in the form of carbon dioxide. And then this acetaldehyde, it accepts the electrons from NADH and it gets converted into ethanol. So these are two different kinds of fermentations. Fermentation is the anaerobic process. It takes place in the absence of oxygen. So lactic acid fermentation takes place in our muscles when we do very vigorous exercise. At that time our cells can undergo lactic acid fermentation. And ethanol fermentation is a process which basically is done by yeast. Like if you have made a bread at home using yeast, so that process involves ethanol fermentation. And also ethanol fermentation is a process by which the wines and the beers and etc., all these alcoholic beverages, they are made. So that's all for this chapter, and if you have a question, please let me know.